Welcome back. This is part 3 of IGCSE Biology Paper 6, Alternative to Practical. Today we'll be looking at food tests. In IGCSE Biology, we have about 4 to 5 different types of tests to consider. We have tests for starch. We have tests for reducing sugar, that's glucose. We have tests for protein. We have emulsion tests for fats. And we also have tests for vitamin C. But for this class, our major focus will be on the test for starch, protein, reducing sugar, and lipid. So the procedures are very simple and need to be well understood. For starch, you just add iodine directly to the substance to be tested, and then you look for a color change. So the expected color change is blue-black to show that starch is present. Let me remind us quickly before we move on that you need to know some things about each of these tests. Number one, you need to know the reagent to use. Number two, you need to know the steps or methods required. Number three, you need to know the color change to show a positive result. Okay? So let's go to protein. To test for protein, you use burette tests, and all you need to do is to add burette to a solution that contains protein. The expected color change is purple, or what we call violet. Remember that burette itself is color blue, so it turns to purple. Then the next test, reducing sugar. When we say reducing sugar, we are talking of glucose or maltose. They are reducing because they reduce the color of Berry solution. So Berry solution, when added to a suspension of reducing sugar, will turn red, brick red, if glucose is present if reducing sugar is present in high concentration. But there are steps involved which need to be well understood. You add the solution to the sample, or to the solution itself, and then you have to heat for like 2 minutes using water bath at a temperature of 80 degrees Celsius. So please take note of that. The color usually changes gradually from the initial blue through green, yellow, orange, and eventually red. So it turns brick red if reducing sugar is present in large quantity. So let's talk about the emulsion test for fat. You add ethanol to the solution in the suspension and then shake thoroughly. Then you add water. You look out for color change. If it turns cloudy milk or milky, then you can know that lipid is present. Okay? So please take note of this. These questions usually come so direct in past papers. Let's look at some questions and see what is expected of us. Here's a question that is talking about chicken's egg. And here, we are meant to describe how ethanol can be used to test a sample of food for presence of fat. Of course, we know that egg contains fat and also contains protein as the major food substances. So how do we test for fat? That's the question. And we have to just describe the same test we have known, the emulsion test, this way. So you name the test, which is emulsion test. You talk about the mixing of the sample with ethanol, and then you talk about addition of water. Then finally, you talk about the expected color change. It has to be to form a cloudy suspension to show that fat is present. Any three of these will score your marks, but there are 4 questions there. Sorry, 4 mark points. So let's look at another question also on this topic. So let's look at the same egg. It's a continuation of the previous question. Now, it's to be tested for reducing sugar. The steps have started. You are meant to predict the next step. So, they said A syringe was used to collect 2 cm³ of the sample. The sample was taken, 2 cm³ of various solution, that's equal volume, was added, various reagents, and then they shake it thoroughly in the test tube. The question is, what is the next step? If you remember from the table we just reviewed, the next step is to heat. Okay, now you heat in water bath, and then remember the temperature required or recommended is 80 degrees Celsius. So, but once you mention heating, you get your mark here. Okay. Alright, and the next one is the positive result to show that reducing sugar is present. Again, we've talked about this. Brick red is the one that shows that reducing sugar is present in very large quantity, but if it is in traces, it can be yellow. If it's very few, very little, it can be green, orange, but the one that shows that reducing sugar is present in the highest concentration is red, or brick red. So thanks so much for listening. There will be more to learn in the past papers, so please do yourself a lot of favor by checking out the past papers and try to apply what you've learned so far. Thank you so much for your time. Our next topic will be enzyme activities. Please subscribe and like our channel. Thank you so much.